Thanks. I really appreciate the chance to talk about Oculinea and our drug delivery technology. Oculinea is a preclinical stage company started up out of UCSF from the bioengineering program at QB3. And it's a little bit of an outlier. I'll be showing this platform technology today, but mostly concentrating on glaucoma. But I hope by the end of the talk, I'll be able to convince you that an effective way to treat glaucoma is with intravitreal sustained drug delivery, such that glaucoma will be another back of the eye therapy for us. This is the core technology for Oculinea. This is based on work that we're translating out of the laboratory of Tejal Desai in the Department of Bioengineering, who's developed numerous fabrication techniques and material advances to create these structured, engineered, thin film biopolymers. And these polymers act as a coat or a capsule around a drug core. So this is a solid cylindrical implant. It's injectable, it's bioerodible, and as I'll show you, we envision injecting it into the vitreous cavity or into the anterior chamber. The way the technology is shown here in the electron microscope here, with the techniques that are available, we can structure the porosity and other characteristics of the device shell so that it matches the pore size to the molecular size of the target molecule, whether that target molecule is a small molecule or whether it's a biologic. In that way, the drug is released across the membrane in a single file fashion. So it's this hindered diffusion that gives rise to zero order drug release. That is, the drug is released at a constant rate day after day at the target therapeutics. And this gets away from problems that we have with other, let's say, standard intravitreal injection or some other technologies where you have an exponential release. That is an initial high threshold bolus drug concentration and then a exponential decline over time so that it reaches subtherapeutic tail. By using zero order, we can take a tiny amount of drug and maximize the therapeutic duration with this pharmacokinetics. As I said, this is a platform technology, so this is previously published work in a feasibility study with Regeneron looking at their drug aflibercept. And you can see here, and these are in vitro studies in pH and temperature controlled fluid chambers, and you can see that we achieve near zero order release of this biologic drug. And importantly, for these delicate, complex, large molecules, we have good structural stability of the drug out to almost 3 months here, and structural stability for antibody-based drugs or chimeric proteins like this is in terms of keeping it in a monomer form so it doesn't form aggregates of dimers and trimers. I'll move on now to the lead program with Oculinea, which is the glaucoma MIDS, MIDS standing for Miniaturized Injectable Delivery System. And you can see a picture of the device here. It's a small cylindrical solid implant. It loads into a 23 gauge needle or a 25 gauge needle. And again, in the in vitro studies, you see that we have near zero order release over 6 months with this small molecule. It's a beta blocker agent with those pharmacokinetics reaching zero order over 6 months. Can we hit the video on that one as well? This is modeled very much on the Ozurdex pellet, and I think the Ozurdex is the most widely used intraocular drug delivery system. I use it in 5 or 10 patients every day. And so in the form factor, the scale, and the injectability of this is very similar to what most of us are familiar with this established procedure. So I'll show the preclinical animal data now. This is using a rabbit eye model. And here we're comparing injection of the device into the intracameral space, which makes sense for a glaucoma device, versus injecting it into the intravitreal space. And in Oculinea, we think there are some real advantages for this to be an intravitreal injection of the MIDS device. First advantage is intravitreal injections are what we're all comfortable with. It's easier. You can almost do it with your eyes closed. I can teach a resident how to do it in 5 minutes. And the second advantage is we can stay away from that delicate tissue in the cornea. Some other implants that have gone into the anterior chamber space have had issues with corneal endothelial toxicity that has limited the repeat usage of the device. We think that if we inject it into the vitreous cavity, there will be an opportunity to use the device over and over again with repeat injections to treat chronic diseases like glaucoma. And then the final advantage is, especially for the mechanism of action of this class of beta blocker drugs, our target tissue is there at the ciliary body, as shown in blue. And so a device that's in the vitreous cavity will have a higher concentration just by proximity to that target tissue. And I'll show you some HPLC data to support that. That's shown here, and I'll just draw your attention to the columns that are at the target tissue in that ciliary body, and you can see that shown in red, there is superior drug level penetration into the ciliary body from the intravitreal injection compared to the intracameral injection. No big surprise, it's closer. Similarly, we have higher levels of the small molecule drug in the vitreous as well. An interesting thing in the rabbit studies, and I think for intraocular approaches to glaucoma with this class of drugs overall, is there is no detectable systemic levels of the drug in the blood of these rabbits. And this is important for beta blocker drugs that can, have rare serious complications on things like heart rate and blood pressure. So it might be something that improves the safety for this class. We've also looked at the ocular safety, both with weekly clinical exams where we didn't see any adverse events associated with the drug or with the device, particularly no intraocular inflammation. And similarly, over 2 months, we don't see any histopathologic abnormalities with the use of this device. So I'll say thank you there. Thanks for the chance to talk about this. This is a company where we're very pragmatically approach to a product and delivering something into phase 1, 2 clinical trials within a year. Thanks very much.